Hi, my name's Leighton and I'd like to share some work I and my colleagues Angelika Kiepas and Peter Koch have done, benchmarking overall genome relatedness index methods. Average nucleotide identity, or ANI, is a measure of how similar two DNA sequences, usually genome sequences, are to each other. ANI is one of many overall genome relatedness indices, or OGRI measures, and ANI is usually interpreted to be the true proportion of identical sites in each genome being compared. But does it actually measure what we think it does? This question is important in part because ANI is a key measure that's often used to assign microbial taxonomy, to identify pathogenic bacteria, and to contaminated material. If two genomes share more than 95% ANI with each other, they're often taken to be members of the same species, although there's disagreement over the choice of threshold and whether a threshold is even appropriate. Classification using ANI has been the basis for some controversial reclassifications of important and significant bacteria. This being the case, you might think that there's been comprehensive, objective benchmarking for all of these ANI methods that demonstrates they measure the true difference in sequence between genomes accurately. This has not, however, been the case. The first ANI method to be proposed, ANIb, which uses BLAST-N to compare genomes, was shown to broadly agree with the results of DNA-DNA hybridization, or DDH. But DDH experiments are highly variable and do not directly measure sequence identity, so the true values were unknown. This initial description of ANIb, therefore, did not establish the accuracy of its measurements of genome sequence identity. Later ANI methods, such as ANIm, fastANI, and OrthoANI, were benchmarked against ANIb to show that they generally agreed with that method. But this kind of transitive benchmarking of ANI methods against another ANI method whose own accuracy is not established is only a form of consistency test, not a validation of the accuracy of each of the methods. Recently, the sourmash tool was benchmarked against a synthetic ground truth, as well as against the OrthoANI method, but this kind of objective benchmarking against an actual ground truth is unusual to see in the literature. As part of the NSF Bio BBSRC funded Genome Archive project, we wrote a tool called pyani-benchmark that generates synthetic bacterial genome sequences for use as ground truths when assessing ANI or OGRI methods. We used this tool to generate a number of pools of 100 synthetic genomes under the influence of a number of operations that mimic evolutionary changes to genome sequences. These changes included random point mutations at high, medium and low rates, and three kinds of structural variation: inversion, recombination and shuffling. The sequences in each pool are the same length and evolve from a single seed genome over a series of generations, with duplication of genomes at each generation. As a result, bases at any single position in each genome are evolutionarily equivalent, and we can calculate an absolute ground truth for the percentage identity difference between any pair of genomes. In this way, we established over 350,000 ground truth pairwise comparisons for benchmarking, incorporating a range of sequence distances and a variety of structural variation. This allowed us to precisely estimate the accuracies of multiple ANI or OGRI methods, the range of identities over which they're accurate, and whether and how method performance is influenced by structural variation. We examined the performance of 4 OGRI methods implemented in our software tool pyani+: ANIb, ANIm, fastANI and sourmash. All were run with default settings. ANIb and ANIm are alignment-based methods. fastANI and sourmash use k-mer-based sequence comparisons. We test these methods first in the absence of any structural variation. The only process acting on the synthetic genomes in this case is point mutation. The first result shows the level of agreement between each method and the actual ground truth. We plotted the ANI method estimate on the y-axis and the ground truth on the x-axis in each case. The orange diagonal line indicates the expected result if the method accurately estimates the true ground truth. In the upper left figure, ANIb results track the orange line quite well between about 80 and 100% identity and the ground truth before beginning to deviate. In the upper right figure, ANIm results also track the orange line well between 80 and 100% identity, but fastANI in the lower left figure looks to consistently underestimate the ground truth identity over the same range. Sourmash in the lower right, appears to be a good central estimator of the ground truth, but individual results show quite a bit of variation about this central estimate. ANIm and ANIb appear to perform very similarly, but we can examine their performance more closely. In this plot, we show the deviation from the true pairwise identity on the y-axis and the ground truth on the x-axis. Results for ANIb are shown in blue and those for ANIm in yellow. Two things appear obvious. Firstly, the deviation from the true pairwise identity is typically much larger for ANIb, up to about 0.2%, than it is for ANIm, less than 0.03%. Secondly, the ANIb results are actually two sets of results, one of which is much less accurate than the other. We can make a similar plot to show the effect of introducing structural variation into the synthetic sequences. Here the y-axis shows the change in ANI estimate when structural variation is applied to the genomes being compared. Now we can see that the ANIm estimates, in yellow, are all found along the orange line indicating agreement with the ground truth. Structural variation has no effect on ANIm results. However, the blue data points, the ANIb comparisons, spread around this central orange line, indicating that ANIb estimates are more strongly affected by structural variation. I hope you enjoyed this introduction to our work. We didn't have time here to look at all the results we show, so please do have a read of our poster to find out more, including how the performance of all of these methods changes when structural variation is a factor in genome evolution, and how well each method estimates alignment fraction, also known as genome coverage. If this kind of work interests you, please do check out and use our software, pyani+. We think it's really good, and we'd like you to try it out for your own work.